Hello everyone, we are the Group 5.
And for today's activity, our objective is to know the different physical properties of leaves. The length of papaya leaf consists of 7.2 inches and the width is 6.13 inches. The length of tobacco leaf consists of 3.7 inches and width consists of 3.13 inches. The length of ampalaya leaf consists of 3.3 inches and the width of ampalaya leaf consists of 3.8 inches. The length of okra leaf has measured 4.2 inches and the width has measured 4.14 inches.
Under a microscope, a papaya leaf would likely appear similar to other plant leaves, with a network of veins, chloroplast containing cells, and possibly stomata for gas exchange. Additionally, papaya leaves might exhibit unique characteristics such as papaya-specific leaf shapes and structures. When observing a papaya leaf under a microscope with diluted hydrochloric acid, HCl, You might see similar changes as with other plant leaves, such as alterations in cell structure, breakdown of cell walls, and the release of intracellular content. This could also reveal any calcium oxalate crystals present in the leaf tissue.
As we first saw the result, we are amazed because of the color that looked like a lava, brownish yellow, and also we saw that there is also a black dot. At the edge of leaf, there is a small amount of bubble and a color of dark orange and black.
a microscope, the ampellia leaf would likely appear as a network of veins surrounded by cells with distinct shapes and sizes. You might observe chloroplasts, which give the leaf its green color, and possibly structures like stomata for gas exchange. Additionally, you might see any hairs or trichomes on the leaf surface. When observing an ampellia leaf under a microscope with diluted hydrochloric acid, HCl, you might see changes in the structure of the leaf cells due to the acid's effects. Include changes in cell shape, the breakdown of cell walls, or the release of intracellular contents. It might also reveal any calcium oxalate crystals present in the leaf tissue. Under a microscope, an okra leaf would likely display a network of veins, with cells containing chloroplasts responsible for photosynthesis. You might observe distinct cell shapes and sizes, along with any trichomes or hairs on the leaf surface. Additionally, you might see stomata for gas exchange and other structures typical of plant leaves. When observing an okra leaf under a microscope with hydrochloric acid, HCl, you might see more pronounced changes in cell structure, such as further breakdown of cell walls and potential alterations in cell morphology. This could provide additional insights into the leaf's cellular composition and response to acidic conditions.
According to Google, the microscopic examination result of a dry marijuana leaf under a microscope reveals trichomes, which contains cannabinoids like THC and CBD. You may also observe plant structures such as stomata, glandular hairs, and the overall cellular arrangement. In conclusion, when we apply HCl to the material known as adulterants, it performs differently. The result shows how the leaves have been damaged by HCl. We come to the conclusion that each leaf has a distinct form and appearance under the microscope.